BIOL 100, Chapter 36, Sensory Systems. So this chapter actually is mostly focused on human sensory systems. There's obviously other senses that we might not talk about, like electroreception and magnetoreception, that would be more appropriate for certain animals like sharks, which is like my go-to, so I wish I could talk about it more in here, but we are human biology, so we're going to be focused on that. And we're going to be talking about how we sense stimuli and how that goes through. We talked a little bit about nervous systems last week, and so you kind of understand what happens in terms of action potentials and sensory pathways, but we're going to go a little bit more into detail today. But in order to sense something, we've got to convert a stimulus, right? Like a light, a sound, position of your body, pain, anything like that. And we've got to convert that into an electrical signal within our nervous system. That process is called sensory transduction. Types of sensation. So we have 5 actual senses as humans, right, that we really talk about. Olfaction, which is smell, gustation, taste, equilibrium, balance, and body position, or touch, you could say. Vision, hearing, and then, like I said, there's these other senses as well, electroreception, magnetoreception, which we largely don't really have. And then going into touch a little bit further, we have different types of somatosensation. So somato being like touch, basically. We have vestibular sensation, which is spatial orientation and balance. We have proprioception, which is the position of bones, joints, and muscles. And then kinesthesia, which is limb movement and tracking. Okay, so how do we actually go through the sensory transduction pathway? How do we sense these different things that are happening around us? First, there's got to be a stimulus, right? Whether that's a light that we're seeing, or we're hearing something, or we're feeling something, there's some sort of stimulus, and we have to be able to receive that. So we have some sensory receptors, depending on what sense it is, that's activated because it's receiving that stimulus. And that is going to lead to ion channels being affected, which will change the electrical potential. So if you remember, as we change the electrical potential, as we make it more positive, our membrane is depolarizing, and that is going to lead to an action potential. If there's a negative change, it's hyperpolarizing the neuron, so it's kind of going back to its original state probably. But if polarization change is sufficient, so if we cross that certain threshold that we need for an action potential to fire, then the neuron's going to fire, and then it's going to send off the whole sensory transduction pathway. So just kind of an example of this, and you can look through your book at these examples as well and read a little bit more about them. But so we can see a membrane here that's receiving a stimulus, and again, once it receives it, it's sending the signal down. These ion-gated channels are opening, depolarizing the membrane, and then we are getting the action potential to fire and tell us, hey, there's a certain stimulus here. And this one in particular is looking at within the ear, which we'll talk about the auditory system in a little bit. Okay, so encoding and transmission of sensory information. So this depends on the type of stimulus. It depends on the location of the stimulus within the receptive field, the duration of the stimulus, and the relative intensity of the stimulus. Intensity is encoded by your rate of action potentials, which is the frequency. So how many action potentials are you having? And then the number of receptors activated, which is the population. So this is what tells us how big of a stimulus this is, or gives us an idea of where it is. Is this really painful? Is this just a light touch? Is that something really big in the sky? Is that something small? These are the kind of things that we're encoding when we get this information. In order to perceive this information, we have to go through our nervous system, right? So with exception of olfaction, all of our other senses are, they're routed through the thalamus. And the thalamus is kind of like our brain's processing relay center, basically. And then that goes into final processing regions within the cortex of the brain that are responsible for processing different sensory information. Like we have our somatosensory processing, auditory processing, the occipital lobe, which is the visual processing. But the thalamus is kind of sending those sensory transmissions to wherever they need to go. Which is crazy to think about all this happening in like microseconds, basically. It's happening so quickly. Okay, so up first, we'll talk about some somatosensation. So this is like touch and things like that within our skin. And mammalian skin, including ours, has 3 layers. So we have an epidermis, a dermis, and then a hypodermis, which is a little bit deeper. Okay. And you can see some of these cells in here as well, and we'll talk about a little bit more, but you've got sweat glands, lymph vessel, oil gland, up here, hair follicle, hair sticking out, blood vessels within, and then we're getting into the fatty tissue, into the hypodermis. But the epidermis is kind of the outer layer that I'm talking about here. And we have different receptor types that exist. So we have thermoreceptors that are sensing, different temperatures. Pain receptors, or nociception is what pain reception is often called. Chemoreceptors, and then mechanoreceptors. Mechanoreceptors kind of going to be our main focus for this section right now. Mechanoreceptors just meaning that anything that can sense touch or movement, essentially. And we've got lots of different types of mechanoreceptors. So we've got tactile, and we've got quite a few of those: Merkel's disc, Meissner's corpuscles, Ruffini endings, Pacinian corpuscles, Krauss end bulbs, which we'll talk about all those in a second, proprioceptors, and baroreceptors as well. Okay, so receptor types in the skin, like I said, we've got those different somatosensory mechanoreceptors, and we've got Merkel's discs. These are unencapsulated, and they respond to light touch. So you'll see these a lot of times like in the layers of skin near your epidermis. So like the hairless skin that you have on your palms, your fingers, soles of the feet, lips. These guys are, again, responding to really light touch. And that's the way that you can really pinpoint the location of a stimulus. So they have really well-defined borders for their receptive fields that make them really sensitive to edges and makes it so that you can do things like type on a keyboard. Right? And then we have Meissner's corpuscles. These are also known as tactile corpuscles, and they're found in the epidermis, but they do project into the epidermis. And they also respond to fine touch and pressure, but also can respond to low frequency vibration or flutter. And they can rapidly adapt. They've got really small, well-defined borders, and are responsive to really fine details as well. Not as plentiful as the Merkel's discs and like the palms and fingertips, but you do still have quite a few of them. The Ruffini endings, these guys detect stretch, deformation within joints, and warmth. Pacinian corpuscles, these ones are a little bit deeper. These detect transient pressure and high frequency vibration. And then these Krauss end bulbs detect cold. So the Krauss and the Ruffini endings, both of these are also kind of like a thermoreceptor in a sense. So just an example of a Meissner corpuscle. This is in the fingertip. You're looking at a photo of a bright field light microscopy. And again, these kind of allow for touch discrimination and detail, so like typing on a keyboard and, some of these like meticulous things that we do with our hands and fingers. Pacinian corpuscles, again, visualized using bright field light microscopy. These detect pressure, touch, and high frequency vibration. And that's kind of the basics for the somatosensory system. We don't have, honestly, you could have a whole class on each one of these senses or on sensory biology as a whole. So this will be a pretty quick overview of all of these. But up next, we're moving into the olfactory system. Olfactory is our smell, right? And this is different. It's not routed through the thalamus like our other senses. We have an olfactory bulb here and nerve endings here, and our nasal cavity here. So basically, particles entering into our nasal cavity are essentially hitting these nerve endings, and then that's sending information into the olfactory bulb, which will then travel back into other parts of the brain. But the neurons associated here, if you remember, these are bipolar neurons, and they extend from the olfactory epithelium, which is over here, to the olfactory bulb. So this is what's essentially getting the reception, and then it's sending it into other parts of the nervous system. Okay, moving on to taste. This is our gustation sense. So taste is sort of similar. Olfaction is definitely different out of all of them. They do have specialized olfactory receptors. And now getting into taste, we have taste buds, right? Which are gustatory receptors that are located within the bumps on your tongue called papillae. These filiform papillae are really more for like helping your tongue move stuff around. And they don't have taste cells, but the fungiform papillae, which are up here, which are like the anterior 2/3 of the tongue, so kind of like the top or closer to the beginning of your mouth, basically. Those ones contain about 8 taste buds, and they also have receptors for pressure and temperature as well. So like if your food's too hot or too cold. And then the foliate papillae back here, those ones have prominent protrusions that you can see on this light micrograph over here. And those ones actually contain about 1,300 taste buds within their folds. And then there's the circumvallate papillae back here. And those ones are like inverted on the back of the tongue. And basically each one of them is surrounded by a groove and contains about 250 taste buds. Something interesting about taste buds too, and all of this is in your book, but taste buds are replaced every 10 to 14 days. So that's very different than, say, like the nervous system, where cells really aren't replaced, if ever, but definitely infrequently. And here they're replaced every 10 to 14 days, and they're elongated cells with hair-like processes called microvilli at the tips that extend into the taste bud pore, so that when food molecules, or something called tastants, are dissolved in saliva, they bind with that and stimulate the receptors on those little microvilli. They're kind of like little hair cells, basically. And so the receptors for tastants are across the outer portion and front of the tongue, outside of the middle area, where these filiform papillae are like most prominent, right here in the middle. So we have five primary tastes. Each one has a different type of receptor, just like olfaction, which we didn't get too deep into, and you can read about it more if you would like. But like olfaction, each receptor is specific to its stimulus or tastant, so what kind of food molecules it's getting. And each of the five tastes happens through different mechanisms that reflect the molecular composition of the tastant. So again, depending on what food molecules you're tasting, depends on how they're actually sensed and what sensory receptors are tasting them. But you can see here these little taste hairs, those little microvilli going into a taste pore. And then there's these transitional cells and a gustatory cell. All of those are working together essentially to receive that stimulus of the food, and tell your brain exactly what it tastes like. So like salty tastants that contain NaCl or salt provide sodium ions that enter the taste neurons and excite them directly. Sour tastants are acids and belong to the thermoreceptor protein family. Um, and so when you bind that sour tasting molecule, it's like an acid basically, and it changes the ion channel and increases your hydrogen ion concentration in taste neurons, which depolarizes them and could send them into action potentials. Sweet, bitter, and umami tastants require a G protein coupled receptor. You don't need to know that much about it. Just know that they're all basically different and they have their own receptors, which is pretty interesting and pretty crazy, honestly. Okay, going into our hearing and vestibular system, talking about our auditory system, um, we're looking at sound waves now. And if you know about sound waves, they are conducted through the air. They also conduct much faster in water, which is, I think it's 4 times faster in water, which is why it's really difficult to understand where sound is coming from when you're in water. But for sound waves, the wavelength here, the amount of space between these two peaks, corresponds to pitch. The amplitude of the wave, so how big the wave is here, that corresponds to volume. So you can see here the dashed line is softer in volume than the one shown in with a solid line. Okay, getting into the ear and how we actually hear. So basically all of the sound waves that are traveling at us, no matter what medium we're in, whether it's air or water, is going to first hit this cartilaginous part of the ear called the auricle, and then it travels through the auditory or ear canal, and it causes vibration of this tympanic membrane or the tympanum. That's our eardrum, right? Um, that's the innermost part of the outer ear. Then the sound is actually going into the middle ear. And in the middle ear, we have three small bones called the ossicles that transfer energy from the moving tympanum to the inner ear. So the three ossicles that we have are the malleus, which is the hammer, the incus here, which is the anvil, and the stapes, which is the stirrup, and is attached to this thing called the round window. The oval window. So each one of these does a different thing in terms of understanding and hearing sound, but basically they collect force and amplify sounds. And so they're a very important part of how we actually hear. And the pressure waves that come from these sound waves, they're striking this tympanic membrane and then transmitting that to these three bones up here. And then the stapes is going to this last one here, the little stirrup one. So the stapes will transfer those over to the oval window, which is the outermost structure of the inner ear. So we went from external ear to middle ear. Now we're getting into the inner ear. And those, the energy from that sound wave is going to transfer to the oval window and the fluid of the cochlea. So the cochlea here is kind of a fluid filled. It looks like a little like snail like structure. And it contains receptors for transduction of the mechanical wave into an electrical signal. And so inside here we have a basilar membrane, and it's basically a mechanical analyzer that runs the length of the cochlea, and it curls towards the cochlea center. So this is where transduction is actually happening. And inside of it is the organ of Corti, which again is the organ of sound transduction, and it's just inside the cochlea here. So going back into that sound wave transduction, again, we're going from the outer ear to the middle ear to the inner ear. And that sound wave is going to travel through, vibrate our tympanic membrane or our eardrum, and then that's going to be amplified by our three ossicles, those three little bones, the malleus, incus, and stapes, and then that is going to be picked up by the oval window over here, which is going to cause pressure against the cochlea, and it's going to move through the scala vestibuli and scala tympani. So you can see how the pressure is actually bending the membrane of the cochlear duct at a point of maximum vibration for a given frequency, causing the hair cells in here to actually bend. And that bending of the hair cells from when these sound waves hit it and vibrate different parts of the inner and middle ear, that is what's actually going to send these action potentials and tell your brain, hey, I'm hearing something, this is the pitch, this is the volume, this is where it's coming from. It's giving you a sense of direction and essentially what are you actually hearing, and then processing that information as well. So the hair cells in the inner ear, they are the same as mechanoreceptors. So they have these little thing called stereocilia emerging from the apical surface, which just means basically there's like little hairs coming out. And they are tethered together by proteins, and those open ion channels when the hair cell is bent toward the tallest member. And then when it bends the other way, it closes it. And so that's what allows these different signals to go through. Vestibular information. So if you hear vestibular, it's usually associated with the inner ear, which can be associated with hearing, but also vestibular, we also think of balance as well. And there's 5 vestibular receptor organs in the inner ear. We have the utricle, we have the saccule, and 3 semicircular canals as well. There's about 30,000 hair cells in the utricle, 16,000 in the saccule, which is below a gelatinous layer, and their stereocilia, or their little hair cells, project into the gelatin. And then within those gelatins are these little calcium carbonate crystals that are like tiny rocks, basically. So those tiny rocks, those crystals, are pulled straight down by gravity, but when you angle your head a different way, it actually causes that gelatin to shift and it bends those stereocilia, those little like hair-like cells, which stimulates the neurons and signals the brain like, hey, your head is tilted, which allows you to maintain balance. So this is the vestibular branch of the vestibulocochlear cranial nerve, and this deals more specifically with balance. And the semicircular canals that we talk about up here, these guys are fluid filled, and they are also arranged in different planes. And basically, their hair cells work pretty similar, that they have a like gelatinous cap on them, and they're stimulated by like driving your car around a corner or turning your head really fast or falling forward. And so you have one canal that's horizontal and the other two at about 45 degrees to the horizontal axis. So your brain processes input from all 3 of these together, and it can detect angular acceleration, deceleration in 3 different dimensions. So this is essentially how we detect acceleration. We keep ourselves balanced, we keep ourselves moving in whatever way that we can to keep ourselves upright or in the position that we really want to be in. All of these, when they stimulate neurons, those neurons then basically go to the cochlear nucleus in the medulla, which you'll remember is kind of like the hindbrain essentially, and it sends more projections into the spinal cord as well as the pons, the thalamus, which is our relay center, remember, and our cerebellum. Um, Connections to the cerebellum are important because that's how we coordinate movements. And then there's also projections to the temporal cortex, and that is what gives us feelings of like dizziness. Sorry, you got to hear my dog barking. Something going by. But yeah, that's vestibular processing essentially in a nutshell. Um, and there's lots of different areas of higher processing depending on whether we're trying to keep our coordination or keep ourselves coordinated and moving the right way, or even if we're sending signals to optic muscles in order to coordinate our eye and head movement. All of that is stemming from our vestibular receptor organs within the inner ear. So your ears do more than just auditory information, which I'm sure you kind of already knew. Okay, now we're going to get into seeing, our vision. So as with auditory stimuli, light also travels in waves, and they are a little bit different, right? The compression waves that compose sound have to travel in a medium like gas, liquid, or solid. But light is composed of electromagnetic waves, so it doesn't need a medium. It can travel in a vacuum. And the behavior of light can be discussed sort of similarly, but a fundamental unit of light, a packet of electromagnetic radiation, is actually called a photon. So there's different amounts of light essentially within this electromagnetic spectrum, and parts that are only visible to us, and then parts that we're never going to be able to see, right? So we have wavelength in meters. You can see here how big it is and what we consider it to be: radio, microwave, infrared waves, visible. So this is our visible light spectrum here, which is what we as humans can actually see, is just even a small part of the visible light spectrum. Ultraviolet, X-ray, and then gamma ray. Gamma ray, I think of like the Hulk, right? Um, so visible light is just between 380 nanometers and 740 nanometers. So those are the colors that we actually see. It's this very tiny part of the spectrum here, and you can see how much other stuff goes beyond that, and what other like humans have a fairly small visible light spectrum in compared comparison to other animals as well, like honeybees. And some animals just have a really crazy visual sense that can see much further beyond our spectrum, but also process it in a very different way, which we won't get into here, but it's worth noting. Okay, so how do we actually see things and process visual cues? Let's first talk about eye anatomy. So our photoreceptive cells in the eye are actually located in the very back of the eye in something called the retina. And you'll see it on a slide here in just a second, and this is in your book as well, obviously. And then the cornea is the front transparent layer of your eye. And you also have something called a crystalline lens. This is a transparent convex structure behind the cornea that has the ability to refract or bend light to focus the image on the retina. And then you have this thing called an iris, which is a muscular ring that regulates the amount of light entering the eye. So you can adjust how much light you need based on the brightness that's outside. Not necessarily even outside, but just in general, how much brightness you're bringing in. And then we have different photoreceptors. So we have these things called rods and cones. Rods are strongly photosensitive and they're located on the outer edges of the retina. So this is like our peripheral vision. We use it in the nighttime as well. It can detect really dim light. So this is like when you're stargazing, right? You're supposed to kind of look out of keep an eye on the corners of your eye, your peripheral vision, to be able to see shooting stars. Because your rods are the ones kind of responsible for detecting visual cues like that. Cones are actually pretty much located in the center of the retina, and these are really weakly photosensitive, and they respond to really bright light. And their primary role is working in the daytime as well as to be able to distinguish and see different colors. You can see here we have the cornea is this outer layer, and then we have this aqueous humor down below it. The iris here again, which is basically allowing more or less light in. And then we have the lens. The retina here is, again, kind of on the back of the eye. And all this other like liquidy stuff in here is what's known as vitreous humor. So the retina is here. You can see as light enters it, it's going to hit this optic nerve. And then we can see the ganglion cells, amacrine cells, bipolar cells, horizontal cells, and then we can see the rods and the cones here throughout. So this is just different layers of the retina, essentially. And then just another close-up of rods and cones. So the outer segments of the rods contain something called rhodopsin. And then you can see the nucleus here, the rod, and again, responding to low light peripheral vision. And then over here we have the cone, that the outer segment contains other photopigments. This is what's responding to color and daytime cues. Okay, so rhodopsin, the photoreceptor that's in vertebrates, has two parts. It has a transmembrane protein opsin and retinal. So when light strikes the retinal, it changes the shape from a cis to a transform. This is basically just a altering of structure. We won't get into too much like how proteins and other things can shift from cis to trans. But just know that basically you're altering the structure of it. And so the signal gets passed to one of those things called a G protein, which is transducin. And that triggers a series of downstream events. And all of that basically leads to closing sodium channels in the membrane of the photoreceptor. And so, unlike most other sensory neurons, which become depolarized by exposure to a stimulus, these visual receptors actually become hyperpolarized and thus driven away from threshold. So they do work a little bit differently, but essentially that's how we are getting our visual cues and transducing. So here's the other figure in your book, going through the transduction of light. Again, light striking rhodopsin, which is on the rod cells, and then going through this whole process and figuring out whether we're going to release this glutamate onto the bipolar cell or neuron or not. And just a few things that aren't really in this slideshow but are kind of worth noting. You do have 3 different types of cones as well. And those different wavelengths, so that's how we're able to see different colors with our cones. And the colors of the visual spectrum run on different wavelengths. So longer ones are closer to like red, orange, yellow, like our kind of, I don't know, warmer colors. And then in the middle, you kind of have like, or in the middle, really, you have like green and blue. And then on the other end, the really short end of wavelengths, you have purple. So there's different saturations, different perception of color, and there's a whole lot that we're not really talking about in the visual system. Your book does go a little bit more in depth, but it does get kind of confusing talking about transduction of light, how you're processing colors, how you're processing retinal information, tonic activity, which is in the retina, and there's lots of different things happening, but basically understand that there's different photoreceptors, mainly rods and cones, that are transmitting information into your visual cortex by way of going through and actually really seeing um, action potentials and working through the whole process.